Introduction. Welcome to today's lecture on cell biology, specifically focusing on the intricate topic of the cell cycle in cancer development. As medical professionals, it is essential to understand the molecular mechanisms underlying the formation and progression of cancer. The cell cycle, which comprises a series of tightly regulated events, plays a pivotal role in the development of this disease. Now, let us delve into the details of each topic that will be covered in this script. In normal cellular processes, the cell cycle is tightly regulated, ensuring proper cell growth, division, and differentiation. However, when mechanisms controlling the cell cycle go awry, it can lead to uncontrolled cell proliferation, a hallmark of cancer. Numerous factors contribute to the dysregulation of the cell cycle in cancer development. It involves alterations in genes that control cell cycle checkpoints, cyclins, cyclin-dependent kinases, CDKs, tumor suppressor genes, and oncogenes. The subsequent topics will shed light on these essential components and their interconnected mechanisms. Cell cycle checkpoints are crucial for maintaining the fidelity of cell division. They halt the cell cycle when DNA replication issues or damage occur, enabling repair or apoptosis. The G1, G2, and spindle assembly checkpoints are pivotal. Disruption of these checkpoints can lead to genetic errors and cancer formation. Cyclins and cyclin-dependent kinases, CDKs, are crucial players in cell cycle progression. Cyclins undergo periodic oscillations and bind to specific CDKs, forming complexes that regulate the transition between different phases of the cell cycle. These cyclin CDK complexes phosphorylate specific target proteins, thereby initiating key events in the cell cycle. Dysregulation of cyclins and CDKs can disrupt these processes, leading to uncontrolled cell division and the development of cancer. Tumor suppressor genes are fundamental in preventing the formation of cancer cells. They act as negative regulators of the cell cycle by monitoring for DNA damage, controlling cell proliferation, and promoting DNA repair mechanisms. Mutations or alterations in tumor suppressor genes can impair their function, reducing their ability to prevent abnormal cell growth. Well-known examples include TP53, P53, which is frequently mutated in various cancers, and RB1, retinoblastoma gene, implicated in retinoblastoma and other malignancies. Contrary to tumor suppressor genes, oncogenes promote cell growth and division. These genes encode proteins that stimulate cell cycle progression and other cellular activities, often leading to uncontrolled cell proliferation. Mutations or alterations in oncogenes can result in their constitutive activation, contributing to the development and progression of cancer. Examples of oncogenes include ERBB2, also known as HER2/NOI, in breast cancer and BCR-ABL in chronic myeloid leukemia. DNA damage and repair processes are intricate mechanisms that safeguard the integrity of the genome. When DNA is damaged due to endogenous or exogenous factors, various repair pathways are activated to rectify the damage. Defects in DNA repair mechanisms can lead to the accumulation of genetic mutations, increasing the risk of cancer development. Understanding these pathways and their dysregulation is crucial for developing targeted cancer therapies. Apoptosis, also known as programmed cell death, plays a vital role in maintaining tissue homeostasis and eliminating damaged or potentially harmful cells. Dysregulation of apoptosis can lead to cell survival despite DNA damage, enabling the growth and spread of cancerous cells. Exploring the molecular events involved in apoptosis and the factors that can influence its dysregulation will provide insights into potential therapeutic strategies for inducing cell death in cancer cells. Genomic instability refers to the increased propensity of the genome to acquire DNA alterations. This instability can result from defects in DNA repair mechanisms, abnormalities in cell cycle checkpoints, or alterations in genes involved in maintaining genomic integrity. Genomic instability is a hallmark of many cancers and is strongly associated with tumor progression and resistance to treatment. Understanding the mechanisms underlying genomic instability will aid in the development of targeted therapies and personalized medicine approaches. Epigenetic modifications, which involve changes in DNA methylation, histone modifications, and non-coding RNA expression, can influence gene expression patterns without altering the DNA sequence. Altered epigenetic regulation is a common occurrence in cancer cells and can influence cell cycle progression, DNA repair, and other critical processes. Investigating the role of epigenetic modifications in cancer development is crucial for the development of epigenetic-based therapies. Telomeres, protective structures at the ends of chromosomes, play a vital role in preserving genomic stability and cell senescence. Telomeres shorten with each cell division until a critical length is reached, triggering cell senescence or apoptosis. Telomerase, an enzyme, can counteract telomere shortening by adding repetitive DNA sequences to the telomeres, allowing cell immortalization and facilitating cancer cell survival. Understanding the regulation of telomeres and telomerase is crucial for developing targeted therapies for telomerase-expressing cancers. Cellular senescence refers to the permanent cell cycle arrest that occurs in response to various stresses, including DNA damage or telomere shortening. Senescent cells acquire a distinct phenotype and secrete a combination of factors called the senescence-associated secretory phenotype, SASP. Senescence can act as both a tumor-suppressing mechanism and a contributing factor to cancer development. Exploring the role of cellular senescence and its impact on the surrounding microenvironment will provide valuable insights into potential therapeutic interventions. Given the central role of the cell cycle in cancer development, targeting its dysregulated components represents a promising avenue for cancer treatment. This topic explores various strategies aimed at inhibiting cell cycle progression, inducing apoptosis, or targeting specific aberrant cell cycle proteins for therapeutic purposes. By comprehending the intricate processes involved in the cell cycle during cancer development, medical professionals gain vital knowledge. Understanding cell cycle checkpoints, cyclins, tumor suppressor genes, oncogenes, DNA repair mechanisms, apoptosis, genomic instability, epigenetic modifications, telomeres, cellular senescence, and potential therapeutic strategies enhances cancer research and treatment. Thank you for your attention, and I hope this extensive explanation has deepened your understanding of the intricate connection between the cell cycle and cancer development.